In today's lecture, we are going to talk about the nucleoid and plasmids which are present in the cytoplasm of the bacterial cells. Probably the most important factor on the basis of which prokaryotes and eukaryotes are differentiated is the presence or absence of a well-defined nucleus. What does a well-defined nucleus mean? The prokaryotes do not have their nuclear material contained in a membrane-bound organelle and hence it is irregularly shaped. It is often called nucleoid, nuclear body or nuclear region which is present in the cytoplasm. However, there are exceptions where membrane-bound DNA containing regions have been found in two bacterial genera, namely Pyrrholulla and Gammata. Pyrrholulla has a single membrane that surrounds a region called pyrrhullosomes, which contain a fibrillar nucleoid and ribosome-like particles. The nuclear body of Gammata is bound by two membranes. Most bacteria have a double-stranded circular DNA as their genetic material. Two features of bacterial DNA are particularly distinctive. The first is lack of histone proteins. In eukaryotes, the DNA is packaged by wrapping around histone proteins to form nucleosomes. Such histone proteins and nucleosomes are absent in bacteria. The second important difference is lack of introns. Eukaryotic DNA is interrupted by non-coding regions called introns. Such introns are absent in bacterial DNA. Isolated nucleoids are found to be composed of 60% DNA, 30% RNA and 10% histone-like proteins. The RNA and proteins are required for the efficient coiling and packing of very long DNA in the very small body of the prokaryote. For example, E. coli on an average has a length of not more than 6 mm and has a DNA not less than 1500 mm long. Obviously, the DNA is looped and supercoiled to fit in such a small space. But this looping and supercoiling cannot be random. If it would have been so, replication would have been almost impossible. This suggests that folding is in an organized manner. The DNA is folded to form loops and these loops are held in place by RNA and proteins. As a result of folding and looping, the nucleoid is in the form of supercoiled DNA. The figure here shows an electron micrograph of E. coli supercoiled DNA. Plasmids are defined as extrachromosomal genetic materials, which are capable of self-replicating. The term plasmid was actually coined by Joshua Lederberg in 1952 to describe all extranuclear genetic material. Let us now discuss the characteristics of plasmids. The first and foremost thing is that plasmids do not carry genes responsible for structure and growth of the bacteria. Thus, it should be possible to remove the plasmid from a bacterial cell without affecting its viability. The second important feature is that the plasmid replication is independent of the chromosomal replication cycle. Almost all the known plasmids are double-stranded DNA molecules. Although the first plasmids were circular molecules, some plasmids are also found to be linear. The linear plasmids have proteins attached to their ends like that of the phage genome, to protect themselves from degradation. The size of plasmids can range from 1 kb to 1000 kb. They can harbor as few as 3 genes or as many as 300 genes. They can be of low copy number or high copy number. That is, there can be one plasmid per cell or more than 50 copies of the same plasmid per cell. Plasmids are responsible for many functions like antibiotic and toxin production and metal and antibiotic resistance. We know that the replication of plasmids is independent of chromosomal DNA replication. Plasmids can replicate by two methods: the Theta model of replication and the rolling circle model of replication. Let us first discuss the Theta model of replication. Plasmids are double-stranded circular DNA molecules. The replication starts at a specific site, the origin of replication. The replication proceeds bidirectionally, and as the replication continues, the plasmid appears like the Greek letter theta, and hence the name of this model is Theta model of replication. The green and blue colored arrows represent newly synthesized daughter strands, and the brown strands represent parental strands. The Y-shaped structure formed is called the replication fork, and there are two such forks. Replication continues and two incompletely formed daughter duplexes are formed. These are called catenanes. At the end of one cycle of replication, decatenation takes place, separating the two completely formed daughter duplexes. Since the size of the plasmid DNA is very small as compared to the chromosomal DNA, it requires just 1/10 of the time required for the chromosomal DNA to replicate. Let us quickly take a look at the animation of Theta model of replication. The other model of replication is the rolling circle model of replication, which is also used by some viruses like 5X174. The plasmids have a double-stranded replication origin called ori, which has two inverted repeat sequences IVR. One IVR is the specific binding site for rep protein, and the other is the target site for the nicking action of the rep protein. The rep protein nicks at the specific IVR, creating a 3' end and a 5' end. The rep protein remains bound to the 5' end, as can be seen in the figure. The DNA polymerase binds to the 3' OH end, followed by the binding of helicase, the unwinding enzyme. The polymerase adds nucleotides to the 3' end by reading the intact strand as the template. The 3' end is extended, to which the single-stranded binding proteins bind, and the template appears to be rolling. As the new progeny strand gets synthesized, the nicked parental strand is displaced. When one unit length is replicated completely, the ends are ligated by DNA ligase to form a circular molecule. Complementary strand synthesis of this single-stranded DNA circle takes place to form the double-stranded DNA progeny duplexes. Meanwhile, after the completion of replication of one unit length, the rep attacks the origin again and nicks it to repeat the entire cycle, which continues to give progeny DNA molecules. Let us now have a look at an animation of the rolling circle model. Many plasmids are known today and they are grouped depending on the replication system used by them. All plasmids fall into only one of the many existing incompatibility groups. Two plasmids are incompatible if either is less stable in the presence of the other than it was by itself. Consider a bacterial cell already carrying a plasmid A. Suppose a plasmid B is transferred to this cell. There are two possibilities now. In the first case, both plasmids A and B undergo replication along with the genomic DNA and are passed to the daughter cells on cell division. Here, the two plasmids use two different replication systems and are said to be compatible plasmids. These are unrelated plasmids and are grouped in different incompatibility groups.